Hello, my name is TAMONWAN. I am a fourth-year biomedical engineering student at Mahidol University. Today, I would like to discuss artificial vascular graft from an engineering point of view, including how the structure is designed to sustain a proper blood flow and the fabrication process. Before any further, I would like to talk to you about the vascular graft. So for the patient who are suffering from cardiovascular disease, the physician are going to use vascular graft or more of like a pre-made vessel to bridge between the blood vessel system at the dysfunction area to keep the system of the blood flows going together as usual. And the blockage of this blood vessel can be caused by the blood clot or even the formation of an excessive wall by the fatty acid. However, to bridging this blood vessel, the physician are going to perform the process known as autologous. So commonly this gold standard procedure based before the coming of the vascular graft, the physician are going to take parts of the blood vessel in the patient's body and putting to use as the bridging vessel. So for some of the example, blood vessel can be radial artery or saphenous vein. However, there is a downside for this bridging blood vessel for the new users, since this is due to the limitation of the extensive requirements for the vessel replacement as well as the compatibility that need to be discussed. In contrast, the bypass blood vessels that are being newly deployed may not have sufficient mechanical properties to withstand large stress from blood pressure. So to make it more understandable, meaning that if you using vein as the bypass vessel in place of the artery, this vein vessel has to undergo larger blood pressure, meaning that the wall vessel must receive a lot of blows from all directions. Additionally, the native blood vessel only available in limit range diameter. So if some of the blood vessel with significantly large diameter are being damaged, the physician had to seek for an alternative materials to be used as the vascular graft, which is why this is known as the artificial vascular graft. When designing an artificial vascular graft for specific application, it is best to understand the anatomy of blood vessel as well as the mechanical and biological factors influencing in the design. So as you can see here, this is the cross-section diagram of the blood vessel for artery groups and venous group. The thick laid inner borderline here represent the smooth muscle, and the much lighter one is represent the connective tissue. In the artery group, it has a much thicker muscle layers in contrast to the venous group. However, for in the venous group, it has a much dense connective tissue as well as the internal wall to prevent the backflow of the blood within the system, as well as the assistance of the surrounding muscle to transport the blood with large content of carbon dioxide back to the heart. However, the reason for this distinguished difference in both vessel groups is the fact that both of them have to undergo very significant pressure difference. Since the artery must transport all the blood with oxygen to every part of the body, it requires large pressure to do so. Hence, the system pressure for the artery is ranging around 90 to 100 millimeters per mercury, in contrast to the venous group that only takes 5 to 15 millimeters per mercury. Next is the mechanical factors. There are 2 points that I would like to make. The first one is the stress rate occurrence. Since the vascular graft that are going to be placed in the new area are being subject to cardiac cycle, meaning that there will be a change in the blood pressure constantly. Hence, this pressure are going to act on the vessel wall in every direction. So, this graft are expected to undergo the circumference and longitudinal stress as the fundamental loads required to withstand. Additionally, the butt fold can cause the shear stress within the internal wall as well due to the content that are being transported over the speed. Another thing that I found to be interesting is about the relationship of the elastic compartments that cause the compliant properties within the blood vessel. So here is the graph relationship of the compliance blood vessel based on the physiological pressure. So the physiological pressure are typically ranging from 80 to 120 millimeters per mercury, and the elastic part that I want to discuss is about the elastin and collagen. So these two are responsible for the compliance of the blood vessel, meaning that it's trying to maintain its shape by providing the flexibility over time. So if the pressure is going below the physiological pressure range, the elastin will be responsible in keeping the structure without any further contractions. Since the elastin has much lower load achievable in contrast to the collagen, and the stress that it can undergo is around 2 megapascal. As for the collagen, if it's going beyond the physiological pressure range, the collagen will try to keep the structure without any further expansions, and the stress that it can undergo is much higher by one, and it is 120 megapascal for collagen. So lastly is the biological factor. So this part I want to discuss about the cell adhesion of the endothelium cell. So this is the cell that is responsible for generating new tissue surrounding the vascular graft. If it generates too much tissue in the short term, it could cause the restenosis. It is the condition where the internal diameter of the blood vessel getting too small, leading to a higher flow resistance and causing the blockage in the long run. However, if the vascular graft that are being deployed unable to attract endothelium cell due to the surfacing or etc., it could cause the immune activation by the platelet, forming the blood clot surrounding the internal wall of the vascular graft, which is known as the thrombosis. And this is the worst one because it can cause arteriosclerotic or initial hyperplasia in the long run. Let's discuss about the material for the artificial vascular graft. There are two major materials that have been used in the commercial product. The first one is the synthetic material. It has been used in the commercial product since 1970, and one of which is the expand polytetrafluoroethylene or EPEX and polyethylene tetrapentylate or DRACON. So both of these polymer has been used for the artificial vascular graft in application of the blood vessel with large diameter. However, both of them are still had a downside due to its hydrophobic surface, meaning that it provides a weak biocompatibility that likely to cause a thrombosis afterwards. However, this can be resolved with the surfacing process that are going to be discussed in the fabrication part. Apart from this, there are other types of polymer that have been studied and being applied with other material to enhance the mechanical properties known, and the example is the polyurethane or PU, polycaprolactone PCL. For PCL, this one has been approved by the FDA to be used as the degradable polymer in the implants one. Another one is the polylactic acid. As for the natural material, even though it's able to provide a biocompatibility in terms of the cell adhesion, but it's still weak in the mechanical properties after it undergoing constant loads. So that's why the usage of hybrid material has been studied recently, which is the combination of the synthetic and natural material using both the advantage of both materials to enhance the performance of the artificial vascular graft. And one of the examples is the usage of polycaprolactone (PCL) to provide the mechanical properties in the short term before degradation, and placing with the chitosan to improve the tissue reconstruction after the deployment. However, another types of the vascular graft that actually proved to be useful is the cellular-based material, and these cellular-based material are serving as the fundamental for the tissue engineering vascular graft. These vascular grafts are made from the blood vessel taken from another living and decellularized living with the structure of the non-living cell, providing the mechanical properties in the short term before being regenerated over by the cell within the native living tissues. For the common fabrication process, there are 4 fabrication processes. The first one is erectile spinning, which is like the ideal fabrication process at the moment. It's extrude polymer on the roller under the high voltage electrostatic field. Not only that, it creates extinguished extracellular matrix pattern, but also the even thickness of the vessel. For the braiding, it using multiple materials and weaving into a single vessel. However, this one not only that it provides the nonlinear strength, it also adding the compliance to the vessel itself as well. For the stretch molding, it is an injected polymer that putting in the mold. However, these molds are made of 2 distinguished material, meaning that during the cool down process, it creates this porous pattern that allowing the cell to be seeding and growing over the material itself. Lastly is the 3D bell printing. So it's similar to how the not conventional 3D printing are operates, but there has been the research that able to deploy the cells within the materials along with the polymer to provide the cell differentiation that leading to the creation of the tissue generations. As for the surfacing process, it can be applied to each fabrication directly or even doing it differently. For example, the coating process of extracellular matrix or fibronectin adhering or albumin to provide the cell adhesion and preventing the thrombosis, or even the combination of the erectile spraying with the erectile spinning to improving endothelialization process. In the current research field, there is the way that researchers trying to study to build a better matrix design for the artificial vascular vessel, one of which is trying to mimic the natural blood vessel as close as possible by doing this radial matrix design. So this design are composing of 3 layers: silk, filament, polyacrylamide (PAM), and thermoplastic polyurethane. And these 3 materials allow the researcher to have the types of the vascular that able to mimic the natural arteries when undergoing cardiac cycle. Another part is more of like an upgrade from the current Dragon graft is the longitudinal axis matrix design. So this combination of the Dragon graft putting the uses of the stent modify patterns at the both end of the connectors that will be connected to the natural blood vessel to improving the compliance properties as well as the compatibility. In conclusion, it could be said that there is a plenty room of improvement for the artificial vascular graft in terms of both fabrication and material combination, and it would be best that we able to encourage for more clinical trial to find the best solution for each type of application as well as each individual as well. Here is the reference, and thank you very much for listening.